All right. Good afternoon, everyone. My name is John Servos. I am the Commercialization Education Director for Fast Forward Medical Innovation, and I would like to welcome you to what is our rescheduled first lecture for the Rogel Cancer Center Innovation Program. Today, we're going to hear, and we're very lucky to hear from Shaomeng Wang on the trends in oncology therapeutic discovery and development. You'll hear more about Shaomeng here in a minute after I say a couple words, and we allow everyone to get settled in on the call. So as a reminder, during today's webinar, please mute your microphone unless you're asking a question. We do encourage a lot of questions. So when there is a break in the action or when prompted, you can unmute yourself and ask a question. Or if you would like to use the chat feature or the Q&A feature on today's webinar, you can submit your question there. Our staff will be monitoring both the Q&A and chat function. We'll share additional links and resources as they're brought up. And then, of course, the recording of this presentation and everything related will be sent post-program to everyone and also made available on our website. I've mentioned this on the other calls for this particular program or the webinars, but if this is the first that you've joined us for, I'll just remind everyone that the Rogel Cancer Center Innovation Program is a new initiative between the Rogel Cancer Center, Fast Forward Medical Innovation, which is a unit of the Medical School's Office of Research here at the university, and the Michigan Drug Discovery Program. And there are two major components to the innovation program at the Rogel Cancer Center. There is this lecture series, and then a project-based course, of which I'll describe here in a few more minutes. This is the third. This was technically supposed to be the first, as you know, but will be the third lecture that we've had as part of this series. We've talked about trends, assessing commercial potential, and some preclinical discovery activities related to oncology therapeutics. The final two of the five that we are offering relate to some additional preclinical target and therapeutic development activities, and then the clinical phase of that development. So please, if you've not registered for those remaining two webinars, go over to our website now and you can register there. If you happen to be working on a project, we would be happy for you to join us in this project-based course where you'll receive multi-week mentorship and guidance from commercialization experts. That does begin on March 8th. And that group will meet every other Friday from 2 to 3 p.m. I believe there are 6 scheduled sessions on those Fridays. And ultimately, the goal of that program is to help you prepare a business case to receive funding and to accelerate the development of what it is that you're working on. So more information on that program as well as registration can be found on our website. Okay, with that, I will now turn things over to Managing Director of Fast Forward Medical Innovation, Brad Martin.
Yeah, thanks, John. As John mentioned, my name is Brad Martin. I'm the director of Fast Forward. And I have the honor today of introducing today's speaker, who John mentioned is Dr. Shaomeng Wang. For those of you that have been around Michigan for any period of time, Shaomeng really probably needs no introduction. He's been a prolific investigator here at Michigan. But for the rest of you, he is currently the Warner-Lambert and Parke-Davis Professor of Medicine here at the University of Michigan and the director of the Michigan Center for Therapeutic Innovation. He has a very extensive background. He received his PhD from Case Western in '92. He had stints at NCI as well as Georgetown prior to coming to U of M in 2001. And in 2007, he was named the Warner-Lambert Parke-Davis Professor of Medicine. He's the director of the Michigan Cancer Center Drug Discovery Program and the Molecular Therapeutics Program at the Comprehensive Cancer Center. He has over 320 publications and has spent some time as the editor of the Journal for Medicinal Chemistry. But perhaps more importantly for today's presentation, Shaomeng has over 50 US patents and many times that number in terms of international patents. And he has founded 5 companies. You may be aware of Ascentage Pharma, as well as Oncopia. And Oncopia was acquired by Roivant in 2020. So U of M is very lucky to have Shaomeng. We're lucky to have Shaomeng here speaking today. And so I'll turn it over to Shaomeng at this point. Thanks very much for being here. He's also a hell of a golfer.
Thank you, Brad. I'd like to thank FMI to organize this workshop for the Cancer Center. I think I will share my slides. So
Looks
can you
good.
see my slides?
Yep, looks good.
Okay, good. So I think for today's talk, although the topic was about the trend in oncology therapeutic discovery and development, I thought I'm going to cover maybe 4 different topics. And so this is only 1 hour presentation. And I will try my best to make some key points for each area. Hopefully that we can learn something from these 4 different topics. So the first one is trend in oncology therapeutics current development. I'm going to focus on what has been proved in the last 20 plus years by FDA to try to see that what's the trend in mostly oncology development area. Then I'm going to talk about a few lessons we learned from some of the highly successful oncology drug development in terms what's the take-home message. And I will cover briefly our own Michigan experience in terms of how we develop drug here and what's the route we have taken and what challenging we have met along the way. And also, finally, the impact of new technology on oncology drug discovery and development. And so trying to do my best to finish in the next 45 minutes or so. So the first is the trend in oncology therapy discovery and development. So we did a research looking into what has been proved by FDA, because this is the ultimate testimony in terms of what has been successful in moving all the way from a laboratory all the way to the marketplace, right? So there's a lot of drug in development and many drugs in phase 3 trial, phase 2 trial, and phase 1 trial. So, but just looking at the approved drug in the last 24 years, we have a total of 241 novel drug approved. Among these, we have about 2/3, 155 small molecule based drug, and 86 biologics, including cell therapies like CAR-T therapies. And if we look at that, what the pace of approval, you will see a dramatic acceleration in the last 23 years. And in the 2000 to 2010 decade, we have a total 48 drug approval, basically about 4.5 drug approval each year, which is not a very large number considering that so many companies develop oncology drugs. So this is a very, very small number. And then things are getting better. In the next decade, 2011, 2019, we have 11 drug approved annually, total of 109 drug approved. So this is really double, even more than double what is approved in the previous decade. That's a great trend for people working on oncology drug development. And I'll talk about some of the reasons for that. Really the age of molecular targeted drug and also biologics development. So in the last 4 years, and we have 89 drug approved. So we have now annually 22 drug approved. So this again, essentially double what we have seen. This is only 4 years, so it's not a very maybe highly representative number for like this one for a decade, but the trend is clear. So we really see the pace acceleration of oncology drug development. So approval by FDA. And I think this is great news for patients and also great news for people interested in developing new oncology drugs. And so I think now there's really a lot more opportunity than what had been there before. And so we break it down into what's been proved in terms of small molecule versus biologics. And you can see that for the 2000 to 2013, this is 14 years, there were relatively few biologic approved, maybe on average each year about 1 to 2 approved. And on average, maybe about 4 or 5 for drug approved in a small molecule space. So this is for the 14 years there. Then if you look at the next last 10 years, between 2013 to 2023, we see this essentially very significant improvement in the overall approval for small molecule. And we have some maybe dry years like 2016. But on average, we see many more small molecule drug approved. And another major trend we see that with the biologics. And now in the last few years, we see this number drug approved for biologics actually started to be very, very similar as the small molecule. You can argue also in the last 2 years, the biologics approval has taken over, right? So in 2022, we get 12 biologics. In 2023, we have none. So really, we see right now, bigger pharma and also biotech companies are investing very, very heavily in biologics. So if you're interested in developing drugs for your own target, and consider that not only small molecule, but also biologics. This has become a major trend. So I also look into what has improved in terms of individual drug class for small molecules. And so we see that the drug approved in red, cytotoxic agent, tubulin agents, and also this is another called gonadotropin releasing hormone antagonist. Although the number of drug approved previously, this is very early. This is like more in the beginning of 2000. So there are essentially none of the drug approved in the last decade in this space. So this is really the past, if you look at the trend. And what is noticeable is that in the space of kinase inhibitor, we have actually 82 drug approved. So this was a really golden year for kinase inhibitor drug in the last maybe 15 years or so. That we have 53% of approved small molecule in a kinase inhibitor. But this is also start to change. And so I think that we can say the past 15 years is probably golden age. We cannot necessarily say the next 15 years will be also golden age for kinase because there are many other type of drug approved. So there are other drug approved, including drug in the protein degradation space, including drug in the apoptosis space, so cell differentiation, cancer metabolism, and proteasome inhibition, and PARP inhibitor, and also anti-androgen, anti-estrogen. So there's a number of drugs here. And so this one, I think, give us the overall idea what really has been working in terms of in the regulatory process. And what about the biologics? So biologics that we see a lot of drug approved as a traditional antibody. And so if you include also immunotherapy, which is also traditional antibody, and this actually consists of like 65%. So traditional antibody has been dominating the space of biologics. And we see an emergence. I think that this drug class called antibody drug conjugate. So if you have a favorite target, a favorite area you wanted to work on in developing new therapeutics. So this is one area I think has gained a lot of attention because of success. Another major trend we see is the bispecific antibody, not only just targeting one protein, but targeting 2 different proteins. So you can achieve actually synergistic activity in the clinic. So this is one thing we see in the biological space. We also, of course, see cell therapy in terms of CAR T cells and gene therapy, and also oncolytic virus. And we one approved radiotherapy for prostate cancer and some protein-based drugs. But by and large, I think it's the antibody drug conjugate. So any question here? Okay. So I think that the take-home message for maybe the first part of my talk is that we now see, small molecule space, there's a very healthy growth of the number of drugs approved each year. And many of the drugs are actually molecular targeted drugs. and many of them are in the space of protein kinase inhibitor. And but we also see that in the biological space, there are many more drugs approved each year in the biological space. And there's actually clear trend the biologicals and the small molecule right now is more or less 50-50 in the space oncology drug approval. Okay, so I'm going to spend maybe next 10 minutes or so talk about what do we learn in terms of some of the highly accessible oncology drug development. So we know that people often say that the chance of a drug getting approved by FDA is very low. So if you have a compound entering phase 1 trial, if it's targeting solid tumor, the every chance of getting approval is about 5%. And so that's basically 1 out of 20 drugs after you enter phase 1 trial. And also we know that if the drug is for the treatment of hematological disease, actually the much better chance, that's about 20 to 30% chance of getting approval when you have a drug entering phase 1. So it's a much higher chance, by about 5 times, getting drug approved for leukemia or other hematological disease, malignancy versus a solid tumor. So that's basically that indicating how difficult getting a drug approved for solid tumor. But there's some highly successful example in developing drug for oncology for solid tumor. I'm going to give you 3 examples. Actually, 2 examples in solid tumor, 1 in hematological tumor. Before I start to do that one, I want to talk about what make a good cancer medicine. And so in oncology area compared to medicine for other more chronic disease, efficacy is always the over driving factor in when we consider a good cancer medicine. So in the efficacy space in oncology drug, I think that right now the gold standard is still overall survival, how long we can extend the patient life. And but many drugs get approved with disease-free survival. In some cases, previously you can go to get a drug approved for response rate. I think right now that become less than that. Really, FDA looking for disease-free survival and overall survival when approve a new cancer medicine. And of course, toxicity is also a major factor when drug is being considered for approval. And so we consider basically the risk benefit. So how much benefit will provide the patient, how much risk the patient will endure. So toxicity is clearly one of the major concerns. I mean, the toxicity, there are two factors. One will be the duration, how long the toxicity will last. Another will be the severity, right? How serious the side effect is. Of course, the most serious will be the adverse event, including death. And we have severe toxicity and manageable toxicity. In most oncology drug, we see at least grade 1, grade 2, and in many cases, we see grade 3, grade 4 toxicity. So toxicity in oncology drug is okay, as long as the efficacy is overriding the toxicity. And of course, that for cancer medicine, and we can also think about the patient population. Some medicine is used for treating hundreds of thousands of patients each year, and some of the medicine only treated for few hundred patients or maybe 1,000 patients a year. So there's a clear larger disease and often indication as well. So FDA gave a special consideration for orphan indication in terms of clinical trial in terms of approval. Of course, the cost. Unfortunately, for new cancer medicine, the cost in the US is very high. and typically costs somewhere around $150,000 per patient per year. So it's very, very high cost. But that's the I think the system we have here. We can talk about that in a different time. So what for ideal drug target? And I think when we do research, we all like the target we are working on. We think it's a good target. But I want to really emphasize the ideal drug target, because this will really make the drug development process much more efficient and much shorter in getting approval. And clearly that when we consider drug target, the very first question we're going to ask will be unmet medical need, and where there's a clear unmet medical need in the area you want to develop drug for. And I think that this second bullet point is that whether target is essential for the disease progression. And we know many drug target play a role in different I think phase of cancer development or progression. But many the target are not called essential target. I think in oncology drug development, we call the driver target versus passenger target. But I think we are going to, I'm going to use some example to talk about if your target have an essential role in disease progression, that will essentially give you a much higher confidence in expected predicted efficacy with drug development. Another thing that we need to ask from the very beginning of any drug discovery development will be, what are expected toxicity for the drug target? If you have the drug get into not only the tumor tissue, the disease tissue, but also normal cell, and what will be the on-target toxicity. And inevitably, almost every single drug target have some on-target toxicity, with some exception. I'm going to show some examples. The exception being that the drug target being unique for the tumor cell and not present in normal cell. So this will be a very critical to understand what will be the expected toxicity. And many drugs fail because they have severe toxicity for inpatient before it can reach the efficacious dose or exposure. And it's not because the target is not important for the cancer, but because the target is also important for normal cells. So if you have a target that's very critical for normal cell function, and most likely you're never going to reach the drug exposure required for achieving clinical efficacy. So the basic drug development got to stop. So this is one of the major consideration in drug development. Another thing that we learned for many, over the years is that if you have a clear biomarker, if you know clearly a patient population that you want to go into based upon the biology you learn for your target, that can make your clinical development to be much faster, and also with a much better efficacy in the patient population. And in today's age, in terms of developing precision medicine, this has become very important. It's very hard to develop oncology medicine where you get about 10% response rate in a broad patient population. Then you can get a very large benefit of, overall for the larger patient population get approval. So this has become critically important. I don't have time today to talk about this particular one area, but I'm going to use some example we have to show you that how this actually impacted the drug development. Okay, so importance of good target. And I often think about, for really good medicine, what are the importance, relative importance in terms of target versus the molecule. And we've seen that if you have good medicine, I think that it's critically important to have good target. And also important to have a highly optimized drug molecule with all the things you want to optimize in terms of potency, selectivity, physicochemical property, pharmacokinetics, and the toxicity, particularly minimize off-target toxicity. So this would, the combination of good target and good molecule will likely give you very good medicine. And we also see in many cases, if you have an outstanding target, ideal target, even if your molecule is not ideal, and you optimize the drug molecule for some aspect like potency, selectivity, and maybe PK, but didn't optimize for some aspect like a physicochemical property, maybe even the off-target toxicity issue, you might still have a very good chance of getting a good medicine. So good target is overridingly important for getting a good medicine. And we see some failed drugs, and failed drug including you have a good target but really have a bad molecule for your target. And this actually is very, very frequent seen in development because many companies, particularly biotech companies, they're small, they don't have time to optimize the compound to achieve ideal molecule, to identify ideal molecule. So put a really not ideal molecule into the clinic, thinking they have a good target. But this can also lead into failure in terms of not ideal PK and unwanted toxicity, not due to the target, but due to the molecule itself. So this can also make the whole program, whole drug development program fail. And you can have a failed drug with a bad target, even if you have a highly optimized drug molecule, you can get a bad target with a bad drug molecule, then of course you get a failed drug. So this just to emphasize how important, relatively important, the good target was a good molecule in the drug development. I think often we do not think about this per se. Okay, I'm going to very quickly go over just a few drugs we know very well, just to make a few points. I think many of us in oncology drug development, we know this story, Gleevec. I just want to review briefly the history of this molecule. And this is Gleevec, also called imatinib, is developed by Novartis. And the key player, including Brian Drucker from Oregon Health and Nicholas London from Novartis, who lead the program in developing Gleevec. And this is a highly successful cancer medicine. And while it's not a cure for chronic myeloid leukemia, it's highly effective, and many patients stay on the drug for more than 20 years. So, and by commercial standard, this is very successful as well. You get a peak of sale $4.6 billion in 2012. Why this is a good good drug target? So BCR-ABL is the target for Gleevec, and in CML, we know 90% of CML patients have this called Philadelphia chromosome that is create a fusion protein called BCR-ABL. Basically, this is two proteins fused together, create a new protein, and this new protein is really driving the initiation and progression of chronic myeloid leukemia and also some acute lymphocytic leukemia. And so this target is the driving oncogene for the disease. And this target is also unique for the tumor cell. In this case, Philadelphia positive CML and Philadelphia positive AL. And so this causes the God-given target for this disease. It's very, very rare, and it's not happening every day for every single cancer. And so the drug Gleevec is a acceptable drug molecule, is not highly optimized for this target. And the data do screening in terms of the compound really hitting the target, but not by design. It's also hitting other kinases beside BCR-ABL, also hit ABL, which is a kinase without fusion to BCR, and as well hitting other, including CK, the FLIP3, as well other targets. So it does have other target. It's not really highly selective compound. It's not a highly optimized molecule as well, but this is a highly successful medicine. Okay. So we come back to the few things we trying to think about the ideal drug target and why this is the ideal drug target. And so we know that the clear unmet medical need, and because before that approval of Gleevec, development Gleevec, and no drug was available to alter the natural progression of CML. And this is the first in-class drug change the course of disease and the practice of CML treatment. Okay, so this drug has made the disease from a rare disease to a much larger disease by having a highly effective medicine, which you can actually greatly increase the survival patient. So basically the available patient on the drug has dramatically increased with this highly successful drug. So I think this is really a prime example. You actually create your own market by making a rare disease become a larger disease. And in terms of target play essential role in disease progression, and the efficacy is outstanding. Is in the first phase 1 trial, published in New England Journal of Medicine, 43 out of 44 patients responded. And the 5-year survival has increased from 31% in 1993 to more than 90% in 2023. So you can see the dramatic improvement with this drug in terms of the patient survival. And this also is a wonderful drug because the BCR-ABL is not essential for normal cell function. So the side effect on target toxicity is very minimal. You look at the toxicity, they're all manageable. And you do have some, serious side effect, and some of them I think can be managed, including bone marrow suppression. And I think the liver problem is maybe patient dependent, sometimes dependent upon liver function of the particular patient, and also that in older patient also heart disease is very very common. But in oncology treatment and all this this side effect can be managed if you identify what is the basically co-founding factor for the side effect. And another thing makes this target to be a wonderful drug, we have a clear biomarker. We actually can identify patient based upon the fusion or based upon the Philadelphia chromosome fusion in CML patient. And so this is, I would consider to be a prime example for developing targeted drug. Okay, so very quickly, maybe just for other 2 examples. And this drug actually was discovered by people working in academia, by Michael Jiang and Charles Sawyer working in UCLA in collaboration with David Wang, who founded the motivation for the development of this drug. This drug actually was developed very, very quickly from discovery to approval is only about 7 years. And it's also very successful cancer medicine with a sale of $5.8 billion in 2022. And this is also success for UCLA because they get a little over $1 billion revenue already by selling part of the royalty on this drug in 2016. I think that probably UCLA is getting a couple $2 billion for this drug for the royalties. And why this is a good drug target? And we know that the target this drug working on is androgen receptor is essential for prostate cancer cell growth and development. And AR and AR signaling play a key role in tumor aggression in advanced prostate cancer, in most of them at least. And AR is not essential for normal cell with separate prostate, testes, and muscles. And several AR antagonists already approved before. But the key weakness that Charles Sawyer and David Wang, Charles Sawyer, I think primarily identify for previous drug is that they're not a pure AR antagonist. So based on that idea, they set out to develop really pure AR antagonist, and in collaboration with chemist Michael Jiang in UCLA. So they're really is developing a better medicine, not a first in class, but a better medicine. And this drug was optimized reasonably well, and the compound has a high binding affinity or high inhibition of AR. It's important is the pure AR antagonist versus just a partial agonist compared to previous one. It's orally bioavailable, but it has an extremely poor physicochemical property with the drug. But nevertheless, this has become a highly successful medicine based upon the sales. Okay, so I think that I just want to briefly touch upon the few areas we are talking about for cancer medicine development. Is that this clear had an unmet medical need, and so one important notion is that no AR target agent was approved in previous 15 years before the approval of enzalutamide. And it's not the first AR target agent, but the clear identified the weakness for the previous compound. The efficacy is, I think, good, not as overwhelming as compared to enzalutamide, but in the space of prostate cancer treatment, this is still considered to be significant. And so again, that the side effect is manageable because the important understanding AR's role in normal cell function. And there's a clear patient selection. Is AR positive prostate cancer patient, particularly in the castrate-resistant prostate cancer setting. And so this is a drug molecule with optimized biological activity, but not a pharmaceutical property. But this one, nevertheless, because of the important target, and also that the good biological activity and reasonable oral bioavailability become very effective medicine. So there are other similar compounds developed later, including couple drug approved in the last few years, basically following the footsteps of enzalutamide. So this is another example with a successful drug. Okay. So just maybe a couple minutes on another example called criticinib. And this drug was developed by Pfizer. And this is the target is ALK fusion protein or ALK kinase. And so this was a protein found in some tumor. For example, initially was in non-Hodgkin lymphoma, and later on was also found in non-small cell lung cancer. And with the publication in 2007 of this EML4-LK fusion protein in non-small cell lung cancer, and found to have about only small percent in non-small cell lung cancer patient, but this is a very large disease. So this small percent actually makes this patient population to be fairly large. When Pfizer see the opportunity of developing drug targeting the fusion protein, and of course, that time people understand the success of previous success of Gleevec. So this is really similar to the target of BCR-ABL. And so Pfizer quickly identify a compound that will develop a c-Met inhibitor is also an ALK inhibitor, but with a 10 times weaker potency against ALK. So that's this drug, criticinib. And they put into clinical development. And of course, there was target validation done in terms of where this is a good target in lymphoma as well in non-small cell lung cancer, in cell line model, in animal model, with the activity demonstrated for this drug molecule can achieve tumor regression. And this compound has a suitable pharmaceutical property for drug development, and clearly was not a drug designed on purpose for ALK, was designed on purpose for c-Met. And so this was developed very rapidly. The started trial in 2000, I think 9. And then very quickly in 2011, they get FDA approval with the phase 1 clinical trial. And so there was additional approval for this drug for other biomarker-driven disease like ROS1 positive non-small cell lung cancer. So this is the first in class drug approved for ALK positive cancer with really that everything lined together for this drug target. Okay, so just very quickly revisiting the reason for the success is that we have an unmet medical need so sorry, I have okay, so I think I copied the wrong slide there. So anyway, so I think that I hope that the 3 examples I show you really touch upon the several areas for important consideration for successful drug development. Is that the clear unmet medical need, that the clear relevance of the target playing an essential role in disease progression. And furthermore, there's a clear understanding of the target not really critically important for normal cell function. And we have understanding of the toxicity, on-target toxicity, that a clear biomarker for this drug, as well for the AR positive drug, as well as for the BCR-ABL. So I want to maybe use the next 10-15 minutes to talk about our own experience in terms of what we have done in Michigan. And I give you 3 examples, but those are extremely uncommon example where you have a target like this in front of you. You said, I want to develop a drug. In those cases, and nowadays, that many groups already working on that target, including maybe big companies and small companies. So this is very difficult. So in those cases, and what do you do? And I wanted to use some of the things we have done in Michigan to show that essentially in academia, we can choose a unique development path for what we do. To still have a success and a great success with what we want to develop new medicine. So the point I want to make is that ideal drug target are very few and also highly competitive. And most drug targets are not ideal. They're lacking one of the maybe 3 attributes for the drug development. So I want to give a quick example, actually, how we have done in Michigan. And in the first 10-15 years, we have been doing research here. So we moved here in 2001, and we decided to not work on kinase. We were working on kinase, a number of them. We had a 5-hour grants working on kinase before I moved here. So whether we made a decision to not work on kinase, and but focus on a new research area that in the programmed cell death, apoptosis pathway. We also choose on purpose targeting protein interaction in this pathway for reason that this is, I think, give us a competitive advantage. So we're looking at this pathway. This is a very emerging field of cancer research drug discovery at that time in 2001. And there's a lot of opportunity, and there's a cutting edge research. As no drug ever advanced into clinical development, so the space is wide open in developing new drugs. And also the in terming the target we chose, targeting protein interaction, was considered to be extremely difficult, if not impossible, back in 2001. So really that there were very few companies or labs working on this space compared to kinase, where we know there are many companies working on this at that time, because people see the success with Gleevec, and so they anticipate there will be many other that that can develop. So we also choose to work on this systematically. We're looking at some of the key apoptosis regulator, including the BCL2 pathways. We chose to work on several targets: BCL2, BCX lung, MCL1 as our primary drug target. We also work on another class called inhibitory apoptosis protein, including CIP1 and 2, and XIP. We choose to work on 3 of them. And also we work on how to targeting PVS3 activation by targeting MDM2 PVS3. So we choose a number of targets on this in this pathway for our research, not just choosing on one, because we know that if we choose on one, we may make it a wrong bet. So if we work on multiple of them, we may be able to develop medicine, at least for some of the target. We are not going to be successful in all of them, but we may be successful in some of them. And so what's our rationale at that time? The rationale is that we understand that apoptosis is one of the hallmarks of human cancer. And apoptosis resistance play a very key role in cancer cell resistance to cancer drugs. So we know that if we target apoptosis proteins, particularly anti-apoptosis proteins, we may be able to develop a new drug to kill cancer cell or develop a new drug in combination with existing drugs. And so that's our key hypothesis. And so basically that the biology is supporting our selection of target, and the available biochemical data, structural biology data, cell biology, and animal model also allowed to go after this target. And so that's what the key rationale we use to choose to work on this molecule. So with our research, and we actually advanced nearly 10 compounds into clinical development. So we have advanced 2 molecules in the IP space. We advanced 4 molecules in the BCL2 space. We advanced 2 molecules in the MDM2 space. So really we have advanced a large number of compounds into this into different space. At that time, if you ask me which one will be most likely successful, we really cannot say with certainty, because the biology was all exciting, and we also know so little about the clinical experience with this drug, because no drug in clinical development. And but I think right now that after the more than 15-year research and development, we now have 1 drug targeting IP in 2 global phase 3 trials, 1 already finished. And our BCL2 molecule also in 3 global phase 3 trials. We also have a regional trial finished. Now we are submitting NDA this year. We are submitting NDA also this year for this drug. So at least the amount of, also we have many of the compounds now in phase 2. And I want to give you a brief review about what we learned about this drug in terms of what's the development steps and how challenging it is to develop good cancer medicine. So I mentioned this drug is in phase 3 clinical trial. And so we really at that time take advantage of the structural biology work people have done defining the drug site. Showing here, we did extensive chemistry, identify, I will call ideal drug molecule in terms of potency, in terms of physical chemical property, in terms of pharmaceutical properties. So we know this compound targeting the IP protein with high affinity, with high specificity. So, and then also this compound has very good phase 1 data. And we show that in phase 1, this compound is well tolerated. And we have all the preclinical data supporting the compound could work as a single agent, could also work as in combination with other drugs. But I just want to show you that how long it took us to actually even develop this compound. So, and we nominated the compound in 2007. And we started IND-enabling in 2009. We started phase 1 trial in 2010 here in Michigan. And the phase 3 trial was started in 2020. So 10 years later, after our phase 1 trial, we have a phase 3 trial. And phase 1 of phase trial already finished enrollment. And so the phase 1 trial we did here in Michigan and Mayo Clinic, showing the very good PK for the drug and very good safety profile at the drug. But the one thing we noticed that we see no efficacy as a single agent, and suggesting combination is essential. But we do have a biomarker in patient in terms of the CMM protein degradation, which show us we actually hitting the target. And we see a very good PK profile, very good safety profile. So, and this drug was undergoing a number of clinical trials, including phase 1b/2a trial in ovarian cancer, and in non-small cell lung cancer, in triple-negative breast cancer. We see activity in this particularly TNBC and ovarian cancer, but did not reach the threshold to allow the company to move forward for phase 2 and phase 3 development. And we did a combination with PD-1 and PD-L1 drug in different disease. And then we also did a trial in other one. So success that was seen now is in head and neck cancer in combination with chemoradiation. And this was leading to the global phase 3 registration trial, starting in 2020 and finished in 2023. With expect NDA to be submitted this year. And also there's another phase 3 trial combination with radiation, and another trial going on right now targeting this BCL EGFR inhibitor, and also in head and neck. So the clinical data is actually in phase 2 for high-risk head and neck cancer is really outstanding for this drug. I just want to show you, this is disease-free survival, and this is the standard treatment, chemoradiation. So after 5 years, you have about 75% of patients died, and this is you combine with this drug, our drug. Basically, we improve the disease-free survival by about 2 times, from 25% to about 75%. And so the overall survival also is improved dramatically from 25% to over 50%. So this is something have not seen in head and neck cancer in the last 30 years in terms of this type of improvement. And so this is right now developed by Merck, KGaA in Germany. And so the trial really have done is going to be the definitive for this drug in terms of for the treatment of head and neck cancer. And what do we learn with this drug? So this is not a Gleevec in terms of the very quick development, because Gleevec was approved with the phase 1 data. This drug it will be approved with the phase 3 data. And we do have some of the really nice feature for new drug. It's first in class drug, and maybe a breakthrough medicine for the treatment of high-risk head and neck cancer. And the improvement in overall survival, progression-free survival has been dramatic. It's an unmet medical need, a very large market. It's well tolerated in patients. We're not adding any toxicity to the standard treatment. And because the combination, and we have no clear biomarker for patient selection, that made the clinical development to be very lengthy and very, very costly as well. So this is one of the, I think, lesson we learned that with this type of drug, it just takes much longer than Gleevec to develop. So maybe just very, very quickly in terms of the impact of new technology in oncology drug discovery and development. And I did want to highlight that right now, 80% of the protein is still undruggable or difficult to drug. That's why if I look at all the drugs approved, you see the majority concentrate on very few drug target. And but we have a new technology in terms, for example, right now, the induced protein degradation is the one that we are working on using either a bifunctional molecule or monofunctional molecule, allow to go after those 80% undruggable target. And there are a lot of advantage with this new approach. We can go after undruggable target. We can make the existing drug more efficacious. We can make the existing drug more selective to reduce on-target toxicity, particularly if a target more than one drug target. And with that one, we now have a large number of compounds we develop on the degrader space ready for clinical development. So I will stop here and to see if we have any questions.
Shaomeng, there was one question in the chat from the first portion of your talk regarding trends, and they're asking if you had any comments on trends in RNA medicines.
Definitely that RNA-based therapy is one of the emerging area, and I think that with the success of developing mRNA-based vaccine, and so that's one area people are developing drug for example as a cancer vaccine. So definitely one of the very emerging area right now in developing both RNA-based, mRNA-based medicine, as well as targeting mRNA or RNA directly by small molecule, maybe RNA protein interaction. So that's one emerging area. But I think we see that that's one of the area that is emerging in the space oncology.
Li Lu? Someone else had a question, they have their hand up? I guess not.
Yeah, it's just muted. Thank you, Shaomeng, for the talk. So I just a quick question. You're talking about how to pick the right target for inhibitor. So can you share any experience if we work on the PROTAC or the molecular glue? Any specific we pay attention to select right target for PROTAC?
Yeah, I think the degrader right now, the few things I listed in there in terms of what will be the importance of the target on disease progression. I think that need to be critically defined in every single target. The number two, I think in the PROTAC space, the toxicity is probably amplified compared to inhibitor because we are able to more efficient targeting the target and remove them from the tissue. So versus just simply binding and inhibition. So the toxicity typically much more than inhibitors. So the toxicity is one critical validation one had to do.
Okay, thank you. Yeah.
Anyone else? If not, I had one question for you, Shaomeng. When you were talking about the development experience here at Michigan, you did the phase 1 study here. You saw good safety signals and druggability signals, but you had no efficacy.
Yeah.
What gave you the confidence to go on and say that you have to require a combination and to go on with that phase 2 trial?
Right. I think that's largely based upon our understanding of the target, as well as in preclinical data we have demonstrated. So we know that apoptosis, the target we have, if you're looking at the panel of models, cell line models that are sensitive, it's only about 5% out of hundreds of cell lines we have tested. And so in the majority, 90% cases, you have to use another agent to provide apoptosis stimulation. So we have a lot of data in the preclinical setting to demonstrate that the combination may work. So I think that's largely based upon preclinical data and also understanding the biology. Otherwise, we'll just say, well, it doesn't work.
Right.
And so that's why the company that licensed the drug was able to do a number of combination treatment based upon the understanding biology. So we know from very early on this target, the activity of the drug, the single agent, going to be very minimal.
And obviously the trial did not have that as its primary endpoint, so.
Yeah.
Any other questions? If not, I want to thank Shaomeng for a great talk, for the work that you do here at Michigan. And we will thank you all and say good afternoon.
Okay. Thank you. Thank you, Brad. Thank you all.
Thank you. Bye-bye, everyone.